from Microbe TV. This is Office Hours for Wednesday, June 25th, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you can come and get real science, no lies, no misinformation, no in-your-face lies that everyone knows is a lie, and yet they don't do anything. We'll talk about that today. Tell us where you're from in the chat. We'll shout you out. Meanwhile, I'd like to welcome our moderators for tonight. We have Les. We have Steph SF. We have Barb Mack. We have Tom Steinberg and Vanity Nutrition. It's pretty much everyone. Thanks, mods, for all coming tonight. Really appreciate your keeping civility here in this live stream. So what's on the bill for tonight? We have a bit of news for you, some stories of the past week, and part 2 in the mini lecture on emerging viruses. And of course, we have you. All of you are part of the live stream, and you add great questions and color and chat. So go for it. Don't be shy. Remember, there aren't any stupid questions. Let's see who we have here. Wayne is in Urbana, Illinois, 32C. So let's see what it is here. It's been very hot in New York these past few days. It's just cooling down now. Weather. Yeah, it's 30 degrees. It was up at 36 today. That's Celsius, of course. What's lower temperatures? Tomorrow it's going to be a high of 28, which is 82. It's not bad at all. You know, in honor of the warm weather, I bought a crew neck. I'm no longer wearing my thermal turtlenecks. I have a bare neck, though. I have to get used to it. Right. But it's much cooler. Except at the incubator, it's too cold, and I have to put a jacket on top of it. But once I go out on the street, oh my gosh, the furnace that is New York City. Anyway, thank you for coming, Wayne. Tom from Weston, Oregon. Welcome, Tom. Bird Carver is on the beach where it's 78F. Kang is in Chicago where it is 22C with a chance of rain. Could you interview Pia Volanto, who advised on how to talk to anti-vaxxers? Yeah, I'm going to write that down on my sticky notes that don't stick very well sometimes. Pia Volanto. You know, vole is like to fly, I think. Anyway, anti-vax dealer. Thank you, Kang. Steph is here from San Francisco. Elizabeth is here. Bird is on the beach in Eastham, Cape Cod. Watching a live stream on the beach. I love it. Good stuff. Questella is here. Welcome, Questella from Iowa/Ohio. Rainey Parker is here. Carol, our favorite nephrologist from a hot Bakersfield, California, 35C. And Elizabeth is from West Virginia. Vanity Nutrition is in the house. Welcome. John is in Minneapolis, 19C. That's pretty moderate there. It's good. Mark Martin is in Tacoma, Washington, where it is 19.4 degrees Celsius. He was at ASM Microbe last week. Well, I used to go to ASM Microbe all the time. I really enjoyed it. But there's some issues there, which I will not air, but I'm not going for the foreseeable future. I don't know. Maybe I'll go next year. Nah, I don't know. I just don't know. They get a lot of programming for free, okay? Let's just put it that way. They ought to kick in. But that's just me complaining. I'm sorry, I'm cleaning my glasses. What else we have here? So we have Barb Mac, where it's 18C in the UK. And a dry heat wave. Yeah, you're getting ours, I guess. Future Dr. Vanity is Dr. Vanity in a couple of years. Yeah, she got into a medical school she told us last week. Congratulations. Mark tomorrow records episode 100 of Matters Microbial. Congratulations, Mark. I'm glad you reached this milestone. Keep it up. You got a ways to go to 1,000. But you do 104 a year, so 10 years. Got 9 more years. It's good. Go for it. I love the title Podfather. It's cool. And you got good titles I like. Earth's Virology Professor, Podfather. Will is from Changsha, where it is 27C and humid. I can say Zhao Shang Hao. That's it. Laizi Zhongguo, Changsha. Good morning from Changsha. I don't know why you need all those words. Oh, well. It will remain a mystery. Vanity had another acceptance today, but would have needed to be there in 5 days. I guess you don't want to do that, right? Tom says, arboviruses. Professor Malik's recent lecture at University of Oregon on dual host viruses, where hosts are very different, had me thinking VR could tell us a lot. Yeah, dual host viruses are very interesting. By the way, I'll be going to Oregon next May, almost a year away. Got invited by UOR. So maybe we can do some meetups. Jill is in Kansas. Welcome. Coralie is in Braidwood, New South Wales. Pete is in London. Fairly summery, 18C at 1 a.m. Soul Symphony is in Minnesota, where it's a beautiful evening. You got a tornado watch. Wow. Wow. Gallbladder 88F in sunny and Franklin, Tennessee. Who else watched the day one of the CDC? What is the damned acronym? ACIP. ACIP committee today. I did not. I was working at the incubator. I don't know. If someone could tell us what happened, that would be cool. Maybe you did Gallbladder 8. Maureen, warm and humid Northeast Ohio. Welcome. Few podcasts reach 100, that's for sure. Most of them stop at 12, which I told Mark when he first started, and then he got past 12. I said, good. Christine is in Hershey, PA, where it's 28C. Hello, viruses. Luna, you've been scarce these days. Good to see you. Logotic is in Chicago, 22C. Elena, Privyet, 28C in Reno. I was in Reno a couple of years ago giving a seminar, and I did a TWIV at the university. Yeah, with some local faculty. That was fun. Went to a good restaurant. Jeff is in Elgin, 90F, and had some rain. Gosh. Vincent is back already. Now, wait a minute. Is that bad? You don't want me to be back already? A week goes quickly. A week is nothing in the scheme of things, right? And I count my days by podcasts. And I crank through the podcast, and all of a sudden it's the next week. Monday comes around, you got to crank up for the next week of podcasts. Yes, Gosh. I love gosh. Carl is in Mississippi where it's hot and very humid. And he says it was 41°C in the shade today. That's hot. Viruses Luna found out today that a physician where I work believes SARS-CoV-2 is lab-borne. It came from a lab. He said when he was in the Army, they used to make biological weapons, and he knows they can do it. Well, they didn't make biological viral weapons, I can tell you that, because nobody has ever been able to make one, weaponize one. Now, you can't infect a person with smallpox and send them into a city. That's easy. That's already existing. You have to make anything there. But nobody made biological viruses. So he is not knowing what he talks about. He knows not what he says, to put it kindly. See, I'm smiling. Mike and Lori are in Seattle, 21°C, and partly cloudy. EHS Talent is in Lehigh Valley, PA. Polka Dot Pup, 89F in Denison, Texas. Hot but very normal. When it's hot and no rain, the grass stops growing. I just got an email from Polka Dot Pup. So now I can associate a name with Polka Dot Pup. Virus is Luna. It's hot in Iowa, 30°C. Jeff says RFK Jr. wants to check the antibodies in ostriches that survived avian flu. The horrible authorities want to kill them. What of keeping the birds that survive alive due to natural immunity? Yeah, they want to bring them to Florida. What are you going to do by checking the antibodies? What is it going to tell you? I guarantee you make the same antibodies as in any other animal. It's not about antibodies that do survival. So Jeff says, I'm against these cults. Now, that's a very broad statement. If you have a farm of chickens and they're infected with H5N1, what are you going to do? Just let them live? They die a miserable death. And the virus keeps spreading through the population, and you can't sell any of those chickens. So what's the point? I don't like doing it, but I don't know what the alternative is. Let it rip? They die a miserable death. You want that? Vincent, you were missed and will be missed. Eventually, Mark, they forget about it. They forget about you. And it's okay. I don't require people missing me, but I do like having people to teach. That's all that matters. Carol says, cool study on immune. Yeah, which arms should you get it? The same arm or the opposite arm? The study suggests that the ipsilateral arm, you should do it in the same arm. I mean, I think the difference is small, but it doesn't hurt to get it in the same arm. Actually, it might hurt. You're going to get it many weeks later, so it's not a problem. I just read today. This is very interesting. Where did I read this? I don't remember. I read a lot, so I don't remember. But basically, remember the early myocarditis signature in COVID mRNA vaccines? Okay, which these anti-vaxxers get all worked up about. But in fact, it's treatable. And it's far worse if you're not vaccinated and you get COVID. But anyway, that's not the point. It turns out that when you space out the first and second doses, you don't see any more myocarditis. And we don't see any more myocarditis right now. Interesting. Peter S. is in Boulder, where it's partly cloudy and 25°C. Ms. Blue Ocean, that's the former environmental coffee house. Good evening, everyone from the southern tier of Western New York, where it's 24°C. Hey, how about it? That guy beat Cuomo for the New York mayoral primary. I think that's great. I don't have anything against Cuomo, but this guy ran a really good campaign. You want to know who I'm talking about? All right, I'll pull it up here. I'm going to do my search. I'm going to call it Guy Who Beat Cuomo. And I bet you it comes up. Come on. Zohran Mamdani. 33-year-old Democratic Socialist and current state assembly member. The presumptive Democratic nominee is an energetic campaign focused on issues like affordability, proposing policies like free buses, universal childcare, and a rent freeze. If elected, he'd be the first Muslim and Indian American mayor of New York City. That's cool. Okay, I could talk some more about that, but I don't want to. This is a virus's live stream. If it was my only acceptance, I would have made 5 days happen. I would have done it some way. Yeah, you would have, but it's not, so you don't have to do that. Elizabeth chimes in. The problem is how contagious it is. The culls are the only way to stop the spread. Also, natural immunity in birds is sort of a myth, and it's a horrible death. Jill watched the ACIP ARG. Nothing good can come out of this particular constellation of ACIP members. The ones they had were really good, and contrary to what RFK Jr. said, they had no conflicts of interest. He lied. Furthermore, he has the financial conflicts of interest. This is disgusting. Nobody calls him out on it. Well, many people do, but Congress gave him the okay. Coralie says it's snowing down there, down in New South Wales. Lori M. from Woodlands, Texas, just moved, exhausted, looking forward to chilling and listening to the live chat. Sit back, relax, get yourself a cool beverage, and yeah, we'll entertain you. All right, Jeff, so how do you know if they're going to survive? So let's say you cull all the sick birds and you have these other birds left, and they may just die later. I don't think you can know, and it's a big logistic issue because we're talking about millions of birds here. 47 likes and 117 viewers because FHSCH said, thanks as always. Glad to see quite a few folks on this live feed. It's good, but please hit the like button so you can try to attract other people to come hear us. Artemis, 26C in Montreal, cooled to 30 and humidex of 45. I'll be going to Montreal in just a couple of weeks. Mark Martin, since I had my birthday, I need to get tough with my GP about vaccines for 65 plus. I have a new GP, so that might help. But clearly I need to have a list. You got to have an RSV vaccine. You need 2 doses of Shingrix. You need an annual flu vaccine. COVID is up to you. But for over 65, you could get a variant, a matched booster every year if you wish. And of course, don't forget the pneumococcal vaccines. Why don't you write a letter to Daniel Griffin, Daniel at microbe.tv, and say, Daniel, I just turned 65. What's my bucket list of vaccines? He would love to answer that. And then everyone can learn. GSD Bio, Jonathan is in Encinitas, 20 degrees C. Very cool. It's chill. Eliza from East York, Ontario, 28C and clear. We're really enjoying Tweevo lately. Good. I like Tweevo. Zoran. Yeah, it's cool. Sandy says, Cuomo is a sexual abuser. I'm happy for New York City and Buffalo got a progressive Sean Ryan. Gallbladder 8. with respect to the ACIP meeting. The CDC staffers were excellent, very professional and knowledgeable. Particularly the vaccine safety monitoring team must have been awkward for them, but they were outstanding. Yeah, the first day is them, and then the second day will be the bozos. I mean, Thiomersal? This was eliminated as an issue years ago. What the hell? This is lies that it causes autism. It does not. Ay, ay, ay. We're just in an era of malignant ignorance. Now they're gonna talk about the MMR vaccine. I swear they're gonna do something with that to screw it up and get more cases of measles in the U.S. Alien, I wasn't. Myocarditis mostly with no. Myocarditis was the mRNA vaccines. The vector vaccine was associated with clotting, and that was severe blood clots. No, mRNA. Just think myocarditis, mRNA. Abdul Rahman from Manhattan, welcome. Apparently, Mamdani is giving the Wall Street financiers the heebie-jeebies. They're used to it already with all the tariffs, right? Carl, I had my lymph nodes removed from one side of my abdomen. Does this affect which arm I should get a vaccine in? No, because the vaccine, you get it here in your deltoid, and it's going to go to the axillary lymph nodes underneath in your armpit, which you could, if they're swollen, you can feel them. So it doesn't matter about your abdominal. Most of the vaccine will stay here. I mean, some will spread, but it will get to the other side eventually. But this is the main thing, these axillary lymph nodes. Mark personally does not like the term natural immunity, which sets up people to oppose efforts to help with our immunity through vaccines. Yeah, I don't like natural immunity either. Did we even mention the term tonight? I don't think so. I don't think we mentioned it. But you're just pulling it out to talk about it, right, Mark? It's fine. It's totally fine. Orally missed out on snow by 200 meters, should have walked up the hill. I don't know what you mean. I don't know. What do you mean walk up the hill? Go skiing or what? I don't get it. Myra says, Luna, glad to be back. Been missing a lot of science. Good to see you, Vincent, and still waiting for you in Iowa or Nebraska. Yeah, I said pneumococcal vaccine, yeah. But, Mark, email Daniel and you'll get the whole list. Mario is in Ukiah, Northern California. Do bacteria infected with phages infect humans? Totally. We're full of bacteriophages. As you know, we have a lot of bacteria all over and in us. And they have phages too, yeah. Are they more dangerous? No. The phages control the populations. In many cases, they're lysogenic, so they don't actually kill the bacteria. But no, they have no effect on us, no. Christella got in another fight with an anti-vaxxer in my own house. She started calling me stupid and brainwashed and saying I was chipped. I just completely shut down and stopped talking. Well, this is an anti-vaxxer you're never going to convince. Because, in fact, that person is the one who's brainwashed or not thinking clearly. Hope to never see her again. More ACIP. The public comment segment surprised me a bit. 10 commenters. 2 anti-vaxxers. 1 wanted to end all vaccines. 8 MDs, NPs, RNs, and LPNs extolling the virtues of vaccines. The candidate to head CDC says vaccines save lives. And what was the other thing? What did they say? Vaccine saves lives. And there was another very pithy thing. They don't cause autism. Tdap every 10 years. Yep. Marge 716, 105 in her garden today. Wow. And tetanus every 10 years too. Tdap, right? Yeah, it's the same thing. Do you know any progress on new human Lyme disease vaccines. No, I don't. I don't know what's going on. Let's take a quick look. Pfizer and Valneva have a candidate which is in late-stage clinical trials, phase 3. And they are hoping to apply for FDA approval in 2026. Okay, so next year. Phase 3, it's good. Thanks for asking. I'm up to date on my vaccines, including meningococcal. I need the rabies as I rescue cats. Yep. Yeah, the T and Tdap. Yeah, tetanus, diphtheria, acellular pertussis. Elise Elcio in Belém para Brazil, 32 and rainy. Welcome. Good to have you. Artemis asks, if someone often gets ill with respiratory diseases, does it mean they're more likely to be asymptomatic transmitters? No, I don't see why that would follow, right? People who are asymptomatic have something that kicks in at the beginning. It doesn't have to kick in after multiple infections. So I don't think that having a lot of infections means that you're going to be asymptomatic. John in Port Townsend, Washington, where the weather is cool and cloudy, 55F. MMR vaccine. Mark may have been born at a time when he got a less effective vaccine. Let's see, Mark Martin. How old is Mark? Mark is probably 10 years younger than me. So he was born in 63. And so we cannot assume that he had measles. And we have to find out what vaccine he had. Yeah. Well, Questella, if you have a trashed anti-vaxxer, that's even worse because they're not thinking clearly, right? So just let her sleep it off. Can I share the link for the article regarding the mRNA vaccine spacing and reduced cardiotoxicity? Yeah, I have to find it. I'll find it. Yep. Gilles says the ACIP meeting continues tomorrow. An agenda item is Thiomersal, but unclear what they want to discuss related to this. Do you know? Well, Daniel and I made a guess. There's the only Thiomersal in vaccines is in the multi-dose flu vaccine, right? Instead of a single dose vial, it has multiple doses. So the people have to go in and out with needle, and it increases the chance of contamination, right? So they put their Mayoral in it to prevent that. And I'll bet they want to take it out. Where, as Paul often said, it doesn't make any sense because, what did he say? Since it's been taken out, rates of autism have gone down to some extent, something like that. Anyway, they will probably want to vote to take it out because they're the bogus committee, right? They can do stupid things. It doesn't really matter one way or the other. I just hope that kids don't get a bacterial infection as a consequence. Seems to me he's already made up his mind about autism and vaccines. Of course, years ago, he believes vaccines cause autism, and he still believes the thimerosal causes autism. Sure, absolutely. Mark writes, if someone is worried about the small amount of mercury in vaccines, why would he sell cod liver oil to children? There's mercury in vaccines. I'm sorry, mercury in cod liver oil. So, as you know, Thiomersal is metabolized to ethyl mercury. So I'm wondering what codfish have. Let's look it up. Mercury in codfish has low levels of mercury compared to other fish. It has some methyl mercury. So that's the bad one, right? Methyl mercury. Typically low enough so it's considered safe for regular consumption. Even for pregnant women and children in moderate amounts. The thing is that, after he recommended it for measles, people were not taking it in moderate amounts. Anyway, Lori, a friend of mine, went to the doc for her Shingrix. Hadn't been feeling well, had a rash. Yes, she had shingles. If she had had vaccine appointment a week earlier, one would have blamed the vaccine. Great point, Lori M. Yeah, who would know, right? But these things happen. I like that. Amazing that she had shingles, right? Pete writes, I only realized this week that in the UK, the COVID vaccine eligibility has been shifted from 65 to 75. So that means I drop out. What about, I guess it's still okay for pregnant women, right? I see 200 meters too low. So she was walking too low to see the snow. I got it. Got it. Mark says, no disrespect to anyone with the natural immunity comment. I see that term used a lot, and it confuses friends of mine. Also, yes, I worry about re-upping with vaccines as I get older. Ask Daniel. Nevitz, they need to have a boogeyman du jour to scare the public. The Mayoral, DEI, masks, mRNA vaccines. Christelle, I didn't even know there was a preventative rabies vaccine. I live in the forest. Probably wouldn't be a terrible idea. No, it's not a bad idea. Especially if raccoons come up on your porch regularly. Can Abdur Rahman, can I come visit this week? I think tomorrow would be good. Tomorrow's Thursday. Tomorrow I have TWIM at 2 p.m. So that'll be done around 3. And then I have TWIV at 5. So you could come at, let's say, 3:30-ish. Okay, yeah. Hit the like button, Gallbladder 8 says. We have 151 viewers and 85 likes. Yes, please hit the like button. It's pretty easy, I think. Iris Luna got infected with SARS-CoV-2 recently and symptoms were not bad. But I was very tired, more than having upper respiratory infection symptoms. Many reasons why that could be, right? I'm glad you called it SARS-CoV-2, but you would know. I know, but many people would say I got infected with COVID. So Luna says, an anti-vaxxer will wake up when their kid dies of measles. Well, in fact, the Mennonite in Texas, whose daughter died, they said we'd do it again. We'd not vaccinate again. No remorse. Oh, okay. Jeff mentioned natural immunity in regards to surviving birds. Well, you could make a different word, but it would be longer, right? Infection-induced immunity. It's hard. Someone come up with a better word than natural immunity. I know this crowd can do it. which is not 3 words. Okay, just 2 words. Better than natural immunity. Okay, Corley, the height, I was too low. It snowed on the hill. Sorry I didn't get it, Corley. Thank you for explaining. Obviously, I don't go in the snow at all. Jorge wants to know, why do some phages have genomes much larger than some eukaryotic viruses? There's no reason for that to be a general thing, right? So some eukaryotic viruses are big. They have big genomes. So that's what T4 needs. Let's see. T4 phage genome size. It's going to be 168,000 bases. So vaccinia virus is bigger. So, it's just a matter of how the phage has evolved and what genes it has picked up that are useful for it, right? There's no reason. Size itself doesn't matter, folks. Size does not matter, because you're going to have a tiny genome with a couple of proteins encoded in it, and it's a good virus. It still takes over the host cell. The extra genes are to do specialized functions. Of course, the small virus genomes do not encode DNA polymerase or DNA-dependent RNA polymerase, for example, but the larger viruses do. The adeno, the herpes, the pox viruses, they encode their own. So it depends what you're doing. Now, I know that T4 phages encode their own DNA polymerase, and I think they have a protein that modifies host transcription, right? So, yeah, man. It doesn't. You can't ask the question like, why are they larger than some eukaryotic viruses? It just because you have to take the examples individually, right? Because it's not a general thing, if you know what I mean. Okay. Carol says Tdap every 10 years, but sooner if you're concerned about pertussis, which we're having a nice little outbreak of here in the U.S., right? Ms. Blue Ocean rescues cats. I think I'm done, though. 6, and I buried 2, and there seem to be non-meandering over here anymore. Bad cat. Hello, Bad Cat. Good evening and chat from Winthrop, Maine, where it's overcast and 79F. Mark Martin just turned 67, so he is 5 years younger than me, so he was born in the 50s. And Mark, you are assumed to have had measles, and you don't have to worry about vaccination. It infected just about everyone way back then. Angry Penguin wants to know, how do researchers edit the viral genome for study? Oh, there are several ways you could do that. So, well, first of all, we start with a DNA copy of the viral genome, right? And we can use enzymes that cut out little bits and replace them with a little bit from a different virus. So that would be sort of like editing because we're changing it. So we're basically making a recombinant that way. So you can use restriction enzymes to do that. Or you can use an editing tool like CRISPR-Cas9, where you put in a cell CRISPR-Cas9 with a guide RNA to target it to where you want to go. And then there are various techniques that you can use so that it will edit the genome in the cell, right? And then you take out the DNA and you sequence it and you make sure that it's been edited. And there are other methods too. It's a very common tool in the lab to use. When I was learning, being trained, we used restriction enzymes because there wasn't CRISPR-Cas9. It's a good question. March 716 says the J&J vaccines was associated with VITT, vaccine-induced thrombo, blah, blah. VITT. Let's see. Vaccine-induced immune thrombotic thrombocytopenia. Yeah, it involves clotting, yes. Thank you. Which is an immune-mediated disorder, an excellent example of clinical research at work. Adverse events result in scrutiny and removal of risky therapeutics. The vaccine was withdrawn, but we didn't know about that when we started. Nevitz writes about Lyme disease. International research team finds genetically engineered Lyme bacterial proteins could offer long-standing protection against infection while requiring fewer vaccinations. That's from 2025. So that's in early development. The vaccine that I talked about is in phase 3, so that can't be this. It's too soon. What was an anti-vaxxer doing in your house? Don't let them in again. Yeah, people who get frequent respiratory infections, writes Wayne, might be more likely to continue normal social activity in spite perhaps a few sick days at work. That's right, because they get it so often, they go, hell with this, I'm not taking off work again, and just go to work. Noir LeBlanc, welcome. Thank you for your support of science communication. We really appreciate it very much. Les has a question. If babies avoided CMV via a vaccine, could that reduce inflammation in the elderly? I don't know what you mean by inflammation. I mean, the problems associated with CMV reactivation? Yeah, presumably, because one of the problems with CMV is you get infected, then it establishes a latent reservoir, and then you can't get rid of it, right? So if you could vaccinate the kids before they got infected, you would prevent that, and then it depends how long lasting the vaccine is, right? May not last till your older years. But if it were durable, then yeah, it could prevent reactivations in the elderly. Polka Dot Pup had the pandemic. That's influenza H3N2 in 1968. Since then, I have never been sick, including COVID. I did get 3 COVID vax. Is it possible I had long-term antibodies and that's why I don't get sick? Well, multiple possibilities. So you haven't been sick. You haven't had a respiratory infection, including COVID. So there are a couple of possibilities. One is that you have a great interferon response and you just shut down all infections before they get anywhere. Or, and that may be coupled to the first one. You may get asymptomatic infections and you are actually infected, but you don't know it. Which you could find out by, taking antibody tests for, say, H3N2 or SARS-CoV-2. And then you say, is it possible I had long-term antibodies? It may be that you were a rare person who makes broadly neutralizing influenza antibodies. But, they're not known to be long-lived. It may be that, maybe your T cells are long-lived and that's what's protecting you. So, I don't know. It's hard to know from one case, right? Whether you really haven't been infected or you're just asymptomatic or not. Okay. Artem is right. Someone the other day told me there's no point in wearing an N95 mask because the size of a virus are smaller than the size of the mask mesh. Well, it's true that the size of the virus particle is smaller than the mask mesh, but that's not what you are stopping. When you exhale you're not exhaling virus particles. You're exhaling droplets, respiratory droplets that contain virus particles. Okay, and those definitely get hung up in the mesh of the mask. Okay, so we're exhaling droplets of respiratory fluid containing viruses. No free viruses. Right? They're on the mucosa and they get expelled with a little drop of mucus, for example. So there is a point in wearing it. And it's well known that they can reduce an infection rate. And I wear a mask. I wear a KN95 on the train and the subway. And let me tell you, these days it's not comfortable, but I wear it. Viruses, Luna says they want to develop CMV vaccines to protect primarily immunosuppressed patients after transplant and neonatal CMV. That's right. Because neonatal CMV can lead to birth defects of various sorts. Digicat says, does he really, and he refers to RFK Jr., believe that vaccines cause autism, or does he believe the paychecks he receives for saying it does? It's hard to know. It's just hard to know that someone would want to harm people to make money. It's hard to believe. I think he thinks that they cause autism, and he thinks that there's been a huge conspiracy to cover it up, which just reveals his lack of critical thinking. But he gets money for being anti-vaccine, of course. And so, a big one is HPV vaccine. He works for a law firm that sues manufacturers of Gardasil vaccine for damages which are not related to the vaccine. And that's why he calls Gardasil the most dangerous vaccine ever developed. And it's not. It's completely safe, and it prevents cervical cancer. So why does he want to take it away and have thousands of people developing not just cervical, but for women, but men, anogenital cancers and oral cancers as well? Why? So he can make money? This is the sickest thing I've ever heard, among the sickest things I've ever heard. And now he's in a position of power and can execute this. Oh, and that reminds me, I have some stories, some news, which I never told you because this is supposed to be shouting out where you're from. And I got into it more. So let me stop here and tell you some news, okay? I got to take Digicat off here. This is important. All right, so let me find I have the articles here. So remember, in the past, whenever I quote a New York Times article, I can't open it, but I finally downloaded the PDFs, and now I can show you. Okay, so let's get the first one. This has to do with Senator Cassidy. Okay, let's do a share here. Okay, so this is New York Times. Let's make it bigger. Cassidy in break with Kennedy calls for vaccine meeting delay. He says the members lack experience. So Cassidy, of course, voted to confirm Kennedy. He could have prevented it if he had not voted. He's an MD. He should know better. Has called for a delay in this week's meeting. Of course, the meeting has not been delayed, so all this is just hot air. But let's see what they said. He says here, although the appointees to ACIP have scientific credentials, many do not have experience studying microbiology, epidemiology, or immunology. In particular, some lack experience studying new technologies such as mRNA vaccines and may even have a preconceived bias against them. You think? So Kennedy says they're too close to the drug industry, which is BS. It's a total lie, okay? He defended the dismissals. He called the old panel a template for medical malpractice, and he had a clash with Representative Frank Pallone, Democrat of New Jersey, who was saying, you're wrong. And then Kennedy says, Pallone has taken millions of dollars from drug industry, and he says that the enthusiasm for the previous panel seems to be an outcome of these contributions. And finally, the chair of the committee, a Republican, said to Kennedy, "You have to retract those words because you can't prove them." And thank goodness, I think it's a lady, did that. Subcommittee chairman forced Kennedy to retract his words. I mean, some people have spines finally. So it concludes a discussion of Thiomersal, right? No link has been found to autism, as you can see here, but yet we have to talk about it again. And the presentation on Thiomersal is going to be by Lynn Redwood, the president emerita of Children's Health Defense, a group Kennedy founded and led until he stepped aside. It's an anti-vax group. This is totally ridiculous. So the thing that's crazy here is that when Cassidy supported Kennedy's confirmation, he said Kennedy reassured him of his commitment to protecting the public health benefit of vaccination. They would have a close working relationship, and he would maintain the CDC panel without changes. The mass firing appeared to catch Mr. Cassidy off guard. And he wrote on social media, "Now the fear is the ACIP will be filled up with people who know nothing about vaccines except suspicion." So this is important because this committee, makes recommendations, and that is important for states to make recommendations and also for insurance companies to reimburse them. Cassidy wanted it delayed until the panel is staffed with robust and balanced representation as required by law. But they're not going to do it. So as you know, the meeting went on today. Okay, so that's number one. Just great, folks. Just great. Well, just lies and misinformation all the time. Okay, next. This is an interesting article, which I can share with you. It's in The Guardian. I'm going to share that. Millions of children at risk as global vaccine rates fall. The decline is blamed on health inequalities, COVID disruption, and soaring levels of misinformation and hesitancy. So this is not just the U.S., folks. This is happening in many other places as well. So this is an article that just tries to quantify how many kids are going to be at risk to infectious diseases as a result of decreased vaccination. So measles rates have fallen in 100 of 204 countries. Coverage for at least one dose against diphtheria, tetanus, whooping cough, measles, polio, or TB has declined in 21 of 36 high-income countries, including France, Italy, Japan, the U.S., and the U.K. So this is important, right? Because we're going to have global outbreaks of diseases. Increasing numbers of wild-type polio have been reported in Afghanistan and Pakistan. There's a polio outbreak in Papua New Guinea, where less than half the population is immunized. It's ridiculous. Measles has gone up in Europe. And so, good article. Check that out. All right. And what else do we have here? All right, so this is the article on RFK is grilled by Congress over health spending cuts, vaccine overhaul in USA Today. And he got questions a lot about his budget and merging agencies into this new administration for a healthy America. Frank Pallone, who I mentioned already, cited concerns about his leadership, anti-science agenda. He said the people on most scientists believe Kennedy's views are dangerous and not supported by scientific evidence. I think people are just going to die, Pallone said. And that's when Kennedy said that Pallone was taking money. He says, "You've accepted $2 million and more than any member of this committee," which is a lie. He hasn't accepted that at all. They made him retract that statement. Okay, what else? All right, then the last one I find very interesting. Well, actually, there's two more. Here's one from the New York Times. Kennedy accuses International Vaccine Agency of ignoring science. So, Biden administration had approved $1.3 billion for Gavi, a global organization that helps to purchase vaccines for children in poor countries. Have you heard of Gavi? So Gavi stands for the let's see, what is it? What does it stand for? Gavi stands for Gavi. It must be Global Alliance for Vaccine Initiative, something like that. Gavi stands for Gavi. Okay, anyway, he's canning that support for Gavi. He said no more money for Gavi because they haven't paid attention to safety issues. So he's taken away $1.2 billion from Gavi. He said they haven't paid attention to vaccine safety. That is a lie. Of course they pay attention to vaccine safety. Gavi retorted by saying, we follow the WHO recommendations, which are based on safety. So he's just lying. It's not true that they don't follow safety. And, he says it and he doesn't provide any evidence to it as well. So this is a big problem because, they wanted to raise $9 billion, so they're going to have to make up for that. Now, Gates and others are donors, and so hopefully they can make up for some of that as well. It's really, really horrible. All right, and then the last one here, which is really interesting. It's a New York Times article, but I have it for you. Autism rates have increased 60-fold. This is a guest essay. I played a role in that by Alan Francis, who chaired the American Psychiatric Association task force charged with creating the fourth edition of the Diagnostic and Statistical Manual of Mental Disorders. So he writes, Robert F. Kennedy Jr. is correct that reported autism rates have exploded in the last 30 years. They've increased roughly 60-fold, but he is dead wrong about the causes. I know, because I am partly responsible for the explosion in rates. The rapid rise in autism cases is not because of vaccines or environmental toxins, but the result of changes in the way that autism is defined and assessed, changes that I helped to put into place. So before that, autism was tightly defined and rare. And so he changed that. And you can read more about it if you can get into the article. But once again, I'm glad that these individuals have just spoken out with the truth, because he's just a liar, a malignant liar. Carol says, hope RFK doesn't put Andrew Wakefield on. I don't think he can. He's just too negative. A person has too much, he has too much visibility. Okay, sorry. Patricia asks, was there myocarditis after the Pfizer or only the Moderna? I believe it was both. Nevitz says, I'd be more concerned about the forever chemicals and microplastics everywhere. More concerned than what, Nevitz? Mark just wrote to Dr. Griffin. Good. Thank you, Mark. 1958, because the cutoff is 1957, yeah. And Sandy didn't know there was a rabies vaccine, lives in the woods with critters. all kinds of critters. Mark says visiting the incubator is just awesome, by the way. The incubator is a great space, and I will show it to you and tell stories about it, because I'm full of stories, and I like to talk about it. I'm very proud of the incubator, all the people that were involved in building it. Costella's really on about this anti-vaxxer. Polka Dot Pup suggests acquired immunity. Yeah, but it's acquired if you get a vaccine too, right? Post-infection survival. That's too many words. We need just two. Temporary immunity. It's not temporary, no. Exposure immunity. That's not bad, Carl. I like that. Exposure immunity. Not waning, no. Because it can wane whether you're vaccinated or infected, right? Sharon, in Tasmania, huge snowstorms for 2 days. Welcome. Mark Martin says T4 is a complex variant put together just so. TMV is one RNA molecule. But that wasn't the comparison, Dr. Martin, Professor Martin, Mark. They wanted to know, why is T4 genome bigger than some eukaryotic viruses that infect humans? And for sure, T4 is complex, put together just so, and you need to have all those proteins, as well as polymerases and other cofactors. But some human infecting viruses are bigger. It's just how they evolved, and what they got. Post-infectious immunity. That's good. Yeah, Noir, that's very good. Post-infectious or exposure. Post-infectious is very specific, and it's 2-word. Post-infectious immunity. Let me write that down. I like that very much. Maybe you should have been a virologist or an epidemiologist or a doctor. Post-infectious or microbiologist. I will use that term. Try and promote it. Unless we have a better one. The Piper is from Maine up north. Smelly Pants says natural immunity equals horribly risky and uncomfortable immunity. Infectious immunity. That's what we're going to call it. Wayne says he was born in 52, recently got an antibody assay to confirm I don't need a booster. Asher is in Tacoma, 79, got a COVID booster a couple of weeks ago. Is this good against the new one covering the U.S.? Probably not. Let's see. I just think it's too soon to be updated to the currently circulating strains. I mean, Moderna just got approved for that, but it's too soon for it to get into the chain. Yeah. What if I was born in the 50s and don't remember having mumps or whooping cough? Well, you should get vaccinated against whooping cough. That's what Daniel says. You may not remember having mumps, but if you were born in the 50s, you likely had it. And he forgot to mention on Tweevel last week that I'm enjoying the animal clade guessing game that Nels likes. There's also a plant version. I'm horrible at that one. You should go back to Tweevo and find that game. It's very good. Matteo, family medical doctor in the office watching in between patients. Well, we talked about the meeting. We didn't. This was the first day, mostly CDC presentations. And as someone said, they're highly professional. But tomorrow is going to be the clinker, because there we have mainly this anti-vax guy presenting. A single word for natural immunity, convalescence. That's a real word? Wow. Convalescence. I never heard of that. Let's look it up. Time spent recovering from an illness. Not quite right. I think we're going to go with what is it? Post-infectious immunity. Speaking of masking, I see about 1% of people masking in train stations and subways. Yeah. David, not so sunny West Seattle, working with friends and family to check MMR titers if born between 57 and 68. Surprisingly, mumps has been the most common low titer. 5% of passengers on my flights wear masks. It's probably higher on a flight than on a subway or train, right? Because they're stuck in the same place for so long. How about just immunity? If you get immunity by monoclonal, could it be passive immunity? Yeah, it is passive when you give people antibodies, but we want to distinguish being infected versus being vaccinated. And that's where post-infectious immunity is useful. SRR, thank you for your contribution to science communication. We need it. Thank you so much. Mark says, it would be interesting to see if a CMV vaccine would help cancer, but with RFK, we can only research conspiracy theories. Sharon writes, I got everything. Chickenpox apparently affected my system terribly. Stories from parents and grandparents are useful to remind us how awful it is to have very sick children. And yes, he is against the HPV vaccine too. What's funny is that an entire strain of flu hasn't been seen since the onset of the pandemic. Talking about influenza B? Yep. Neonatal CMV, hearing deficit, and mental hindrances. Bobby says, I hate that virus is smaller than mask mesh argument. Joe Rogan says that, as if one individual virus is just floating around by itself like a mosquito and infecting people can't happen. Okay. I'm going to mark that because we're going to do a mini lecture now. Let's take Bobby off there. And let's go to our mini lecture. We're going to play it. And screen share it. Emerging Viruses Part 2. Okay. Let's close this. Okay. Last time we talked about the fundamentals, right? Now we're going to continue with some examples of emerging viruses. So we didn't know that bats were actually sources of zoonotic infections until 1995, when this paper was published. A number of years later. Bats host major mammalian paramyxoviruses, which is interesting, including Nipah and Hendra, which we'll talk about, but no mention of coronaviruses yet, right? So Hendra virus was discovered in Hendra, Australia, up here. I'm not sure which of these dots is Hendra, but you could look it up. September 1994, an outbreak killed 14 racehorses and a trainer. And eventually they sorted out that the virus spread from flying foxes, those bats that look like they have a face like a fox and a 4-foot wingspan. They shed the virus, and the horses have open barns. The flying foxes fly in and they contaminate the stalls. and then the horses get infected, and then the humans are taking care of the horses, and they get infected as well. The horses continue to acquire infection, and this is a problem because racehorses are very expensive, right? So a vaccine has been developed for horses. The human disease is too rare to have a human vaccine, but if you vaccinate the horses, you will prevent people from getting the disease. So that's a one world health approach. This is, by the way, this blue line is the range of the flying fox. So in theory, this Hendra could be found in all these areas, but it hasn't yet. Now, Nipah virus caused an outbreak in Malaysia in 1998, an outbreak of respiratory and neurological disease on pig farms. And this was associated with 105 human deaths because humans take care of the pigs, right? They culled 1 million pigs. How do you cull 1 million pigs? You dig a big hole and push them in. Unfortunately, that's what they did. And you can find a video of that on YouTube, which I do not want to look at because it's so sad. Anyway, here's Malaysia. And there were many pig farms in the Perak district here, and they were outdoor pig farms. And so what happened was these pig farms had mango trees planted in them, and at night the fruit bats would come and eat the mangoes and excrete virus in urine, drop fruit on the ground, and then the pigs would eat it, get infected, spread it to humans. There were other factors involved as well, I'm going to tell you about in a moment. But this also spread down to Singapore by shipping of pigs. And all of this has now been controlled because they moved the piggeries indoors. So that doesn't happen anymore. But humans have subsequently been infected by consuming date palm sap, right? So here's a palm tree, and they tap it and get sap, and people like to drink this. But it often caused Nipah infections, and they figured out that bats at night like to drink the sap, and they contaminate the sap with virus. And so the low-tech solution was just to cover these date palm sap bottles that are hanging off the trees, and that stops the date palm sap associated transmission. There's a little bit of human-to-human transmission, not very much, not enough for it to become established as a human virus. And we still have infections in different parts of India, in fact. So the factors leading to its emergence, first of a reservoir of fruit bats. In the 70s through the 90s, there was deforestation for pulpwood and industrial plantations that removed the fruit bat habitat. They also did slash and burn clearing, and this haze blanketed much of Southeast Asia. There was a drought caused by the El Nino. So basically, the flying foxes were pushed out of their habitat, and so they went into the fruit orchards. And as I said, they were open piggeries with orchards in them, and that's what attracted the bats. So here's another slide saying basically the same thing: El Nino drought, forest fires caused by us, haze, deforestation, decreasing the habitat, the food supply goes down, the bats move, and they go to the piggeries, infect the pigs, and eventually humans as well. So that's a flying fox, by the way. Doesn't it look a bit like a fox? Long snout, the ears. It's cool, very cool. So here's another summary of Nipah virus spillovers, right? We have the flying foxes foraging in fruit trees on pig farms and contaminating the pigs and also people. Then we had date palm sap mediated infection where the bats contaminate the date palm sap and people eat it and they die. And then people taking care of their dead, they get infected. That's why I say there's a little bit of human-to-human transmission. But even more puzzling, there are some people who get infected who don't have any contact with date palm sap, and we're not sure what's happening, whether there's some flyover event which may be infecting horses and then people. This is a bit of a puzzle. There is now a vaccine in development for Nipah virus. Here's another one, which I particularly like, Junin virus, which causes Argentine hemorrhagic fever. So after the World War II, the Argentinians wanted to convert the pampas to land for the growth of maize. So they used herbicides to get rid of the weeds. They grew corn, they grew maize, but then a new shade-tolerant grass grew in the shadow of maize. And this grass attracted a new mouse, Calamus musculinus, which hadn't been there before. And it had in it Junin virus. And as people went into the fields to harvest, they got contaminated with Junin. And that was a new hemorrhagic fever first described in 1953, Argentine hemorrhagic fever. So it's all anthropogenic activity that's causing these spillovers. Ebola virus. Been multiple outbreaks, of course, starting in the 70s and going through to 2000s. And the size of these circles represents roughly the size of the outbreak. The 2015 West African outbreak, very large, larger than ever. Many outbreaks in Central Africa and one in South Africa here. This is, of course, Ebola virus, a filamentous virus with a negative stranded RNA genome. And there are 4 different kinds of Ebola viruses shown here. And there's Zaire, Sudan Ebola viruses. You can see Zaire is the red. It's caused most of the outbreaks. There have been a few caused by Sudan. And then Thai forest and Bundibugyo are extremely rare. So that's Ebola viruses. It's a filamentous virus with a negative strand genome that encodes multiple proteins. So how are humans infected? This is a classic zoonosis. There's typically, but not always, identified some kind of contact with an animal carcass for hunting purposes, for food. It's then transmitted to other humans by close contact with infected fluids. Short chains of infections. Never establishes itself as a human virus which transmits always. It's always a zoonotic event. So a couple of examples. Gabon, 1996. Zaire Ebola virus, 37 cases. A chimp found dead in the forest was eaten by people hunting for food. 18 people who were involved in butchering the animal became ill. 10 other cases occurred in family members. And then Gabon, 60 cases. The index case was a hunter who lived in a forest camp. A dead chimp found in the forest at the time was infected with Ebola virus. Now, you don't always establish these connections, but the assumption is that it somehow comes from contaminated meat, either non-human primate or even bats. The 2015 West African outbreak, which grew to over 25,000 cases, began in 2013 in this small village here in Guinea, where the first recorded cases, first was a child 2 years of age who had symptom onsets on December 2nd and died on December 6th. We don't know what this child did to get infection, but that child went on to infect many other people. He infected his sister who died. He infected his mother who died. He infected his grandmother who died. He infected a nurse who died. And he infected a village midwife who died. But before that, she went to another village and spread the infection. And then this continued to spread on and on and on. All of this wonderful epidemiological tracing showed how this moved. It's incredible. This kept spreading because of contact in hospitals and burial procedures. So eventually we did infection control and we stopped the spread. So this did not become a human virus. But, even if you have a non-human virus, if you have close enough contact, you will get infected. Now, where is Ebola virus coming from? For Marburg virus, which is closely related, the infectious virus has been isolated from the cave-dwelling fruit bat. And so we are very confident that these bats are the reservoir of Marburg virus. But we've never found Ebola virus, infectious virus, in bats. We have found antibodies in 3 bats. We've found RNA, but not infectious virus. So it's still an open question, and the experts will tell you we don't know what the reservoir of Ebola virus is. But we know that humans, gorillas, and chimpanzees are dead-end hosts. Somehow they get infected, and they don't spread it very effectively. So here we have some scenarios where bats are the reservoirs. Here, the 1, 2, 3, and these are bats transmitting it among themselves, and occasionally they may transmit it to chimps or gorillas or other species. They may transmit it to humans, or these non-human primates may transmit it to humans as well. We just don't know. We do get human-to-human transmission with close contact with infected blood or body fluids like urine, saliva, sweat, feces, vomit, breast milk, semen from someone who is sick or has died. Contact with contaminated needles and syringes, but not by insects, water, food, not by aerosol. The virus enters through mucosal surfaces, breaks in the skin by needles, contaminated needles. You can find virus in the skin, body fluids, nasal secretions, blood, semen. There's an incubation period of up to 21 days during which you are not contagious. You're not contagious until you show symptoms. And what are the symptoms? Systemic prostration, gastrointestinal, respiratory, vascular, neurological, everywhere. It's infecting everywhere, and 30 to 90% case fatality ratio in Africa. Okay, moving on to SARS, SARS-1. This is a post from ProMed Mail, the alerting services for infectious diseases. They used to have a wonderful email service. 2003, from this individual this morning, I received this emails. Does anyone know about this? Have you heard of an epidemic in Guangzhou? An acquaintance of mine from a teacher's chat room lives there and reports that the hospitals have been closed and people are dying. So this is February 2003. Turns out that this outbreak of SARS has been going on since November of 2002. Severe atypical pneumonia in Guangdong down here, but the Chinese authorities didn't report it to the rest of the world until it kind of snuck out on from this chat room in February. 305 cases and 5 deaths in that initial outbreak in Guangdong. Incubation period 2 to 10 days. Chills, headache, malaise, myalgia is the beginning, and then dry cough, shortness of breath, and quite a few people requiring mechanical ventilation. So severe acute respiratory syndrome, very much like SARS-CoV-2. A Chinese doctor who treated some of the first patients in Guangdong, he traveled to Hong Kong in February, stayed on the 9th floor of the Metropole Hotel. He died in the hospital the next day, but he spread infection to 10 people in the hotel who then flew on to Singapore, Vietnam, Canada, and the U.S. before symptoms were evident, and then they started spreading it to other people. Eventually spread to 8,000 people in 29 countries with a 10% mortality rate. But it stopped. Infection control measures and travel control measures stopped it. It never became a pandemic because there was no asymptomatic shedding. Most people started to shed at the peak of illness, and many of them were in the hospital at that time. Very fortunate. I have a travel poster here from Hong Kong, which is from those years. Hong Kong will take your breath away. Life is exciting. So there's the epi curve starting in, here in February, and then peaking, ending in July 2003. And here are the countries, most of the cases in China, Hong Kong, Taiwan, and a handful in other cases in other countries. And of course, this is a coronavirus, SARS-CoV. Where did it come from? Well, if you look at humans here before the outbreak, they don't have antibodies to SARS-CoV, but the early cases were in handlers of animals for the exotic food market, these open meat markets where animals are slaughtered. And these individuals had the earliest cases, and they had a higher prevalence of antibodies to the virus than control groups. So the search went on for the origin, and what was found were highly diverse SARS-related coronaviruses in horseshoe bats, Rhinolophus, in a cave in Yunnan province, about 1,000 miles away from Guangdong. They didn't find SARS-1, but they found all the elements needed to form it by recombination, but they didn't find the direct progenitor. But this pretty much proved that that was the origin. But there were no SARS cases in Yunnan province during the outbreak, right? In that cave where these bats were found, there were no cases in the province. So the progenitor was thought to have emerged in this cave in bats, then infected civet cats, which were then taken to the markets and sold. And these civet cats were found to be positive for virus and for antibodies. So the virus went from bats in the countryside to civet cats to people. The next coronavirus outbreak was MERS, Middle East Respiratory Syndrome coronavirus. September 2012, a 60-year-old male patient died of pneumonia and renal failure. The virus was recovered and identified as a different virus from SARS-CoV, but closely related to bat coronaviruses, and giving the name MERS coronavirus. The virus spreads from dromedary camels. It is endemic in the Middle East and Africa, where these animals are quite numerous, although not every human infection has a camel source, and we don't know what's going on there. But people who own camels or get in contact with them get infection. Camels are all infected with MERS coronavirus at birth, with very little effect on them. This virus doesn't transmit well among humans, so most cases are zoonotic spillovers. There is a camel vaccine in testing, which would be a One Health approach, right? There are not enough human cases, but you vaccinate the camels and prevent the human disease. So here is an older map of the number of cases, 2,500 cases, about a 35% case fatality rate. Most of them in the Arabian Peninsula, but people have traveled and brought the virus to other countries, and that's shown by the blue here. So there is camel-to-camel transmission of this virus. There's camel to human and limited human-to-human spillover, and then human-to-human with no evidence for a camel outbreak, for a camel source. Here, for example, a person traveled from Saudi Arabia to South Korea, and when he got there, then he developed symptoms. He went in the hospital. They had no idea what he had, so they put him in an open ward, and he infected 7 other people, right? Close quarters. Okay, SARS-CoV-2, third pandemic of the 21st century. Starts with a cluster of pneumonia cases of unknown cause in Wuhan. December 2019, the Huanan market here shown was the early epicenter. Most of the cases cluster around the market, the early cases. And of course, over 700 million cases globally and 7 million deaths. Now, here's the Wuhan Institute of Virology across the Yangtze River, and there's no clustering of cases, which would support an origin there. So the data all support an origin at the Huanan seafood market. Now, when the virus was first, the genome was first sequenced and placed in a phylogenetic tree of coronaviruses, you could see that these SARS-CoV-2 isolates formed a separate branch of the tree, and they clustered with a number of bat coronaviruses, including RITG13, 96% nucleotide identity. Now, this was just a genome sequence. Nobody had this virus, and early in the pandemic, people were saying, it was made from RITG13. No, nobody had virus. And anyway, this 4% is 1,200 bases. Couldn't make it from that. But these viruses were distant from SARS-1, as you can see up here. So these are new viruses, which is why they're called SARS-CoV-2. Subsequent to the beginning of the pandemic, many more SARS-CoV-2-like viruses were isolated. In this study, they were isolated from Laos, 4 sites in Laos, and they're called banal viruses. And in fact, many of these have a receptor binding domain that allows ACE2 entry into cells and are very close. closely related to SARS-CoV-2. You can see here 2 lineages of SARS-CoV-2 that emerged in the market is RATG13, and then some of the banal viruses are very close as well. Now, this is a homology map of the entire genome translated into protein of these closely related viruses. All right, so here the comparator is SARS-CoV-2, and then you can see some viruses are very close to it. So the similarity score is 100% at the top here, and then it goes down to 0 here. And you can see that, for example, RATG13 in red, in kind of orangey color, is very similar, and these banal viruses are very similar. But where the similarity drops is very interesting, is in the spike encoding gene. That's right here. So this supports the idea that SARS-CoV-2 is a recombinant of perhaps many of these viruses or ancestors to them. So coronaviruses over the years have come from nature, not from laboratories. We have 1, 2, 3, 4 common cold coronaviruses that originated in animals and perhaps had an intermediary before getting into humans. SARS from bats to civet cats to humans. MERS from bats to camels to human. Is even a pig pathogen, severe acute disease coronavirus. Severe acute diarrheal syndrome coronavirus, which went from bats to pigs, has not spilled over into people. And then we have SARS-CoV-2, where some of the animals being sold at the market, like raccoon dogs, have been found to harbor the genome, but ultimately it most likely came from a bat, because these kinds of sampling studies that I show you that yield highly related viruses to SARS-CoV-2 are from bats. Many years ago, I did a TWIV with Linfa Wang, who has since become an authority on bat-derived viruses. I went down to Australia, in fact, to record and interview him. That's worth listening to. Now, many of these viruses have to be worked at under BSL-4 conditions. There are 4 levels of biosafety, BSL 1, 2, 3, and 4. And 4 is the highest, and you have to work under BSL-4 with viruses that cause high mortality, person-to-person transmission perhaps, and no approved vaccine or antiviral. And we did a documentary at the Needle, which is the BSL-4 facility in Boston. You can find it this link. And this is what you have to do to work in these. You have to put on basically a sealed suit, a plastic suit, and you have an air feed to you so you can breathe. And as you move around the room, there are multiple of these hoses hanging from the ceilings, and you have to unclip and clip them as you move from one place to the other. So how common are these host range jumps that we've given you some examples of? Well, dead ends are very common. We don't see them. Those that make sustaining transmission are probably rare. As we have seen, there have been a limited number of pandemics. They're terrible. They cause a lot of damage, but they're pretty rare. Can we predict them? I don't think so, but we can know what's out there and be prepared with antivirals and perhaps vaccines. We can do early warning screenings in places of high human traffic like airports and train station and shipping ports. We could have made pan-coronavirus antivirals after SARS-CoV-1, but we didn't. Why didn't we? Well, it stopped at 8,000 cases and people thought not important. NIH wouldn't fund any of that work and companies wouldn't get involved because there was no market. Probably could have made a big impact on this pandemic if we had made those, but we didn't. Okay, folks, that's Emerging Viruses Part 2. Let's go back to the questions here. Andy wants to know if prions are the smallest infectious agents. So the molecular weight of a prion is about, I forgot, prion molecular weight. 35,000 Daltons, okay? Keep that in your head. Now we're going to look at a viroid. Let's look at the molecular weight of a viroid because they're pretty small. 85,000 to 130,000 Daltons. And they're like 200 bases of RNA. So prions are the smallest in terms of molecular weight. Yes, the smallest infectious agents. This is interesting. Tom writes, interesting, New York Times is not using the honorific doctor for Cassidy, who has no excuses. He has zero excuses. He wants to keep his job, but that's not an excuse to compromise the health of people. Sorry, bub. Just suck it up and do what's right. Yes, Children's Health Defense was mentioned in booster shots. It was the company that he founded to do anti-vaccine work. Senator Cassidy thinks the members of the committee might not know anything about vaccines. Surprise! Yes, they don't. Visto asks, with so much anti-vax online, should Facebook be classified as a viral vector? In a way, right? You're vectoring misinformation. Mark was in the east end of Long Island. Yesterday it was 34.4 C in the shade. John writes, the lunatics aren't running the asylum, they're running the entire health and human services. Vanity says, Gavi may stand for Give All Vaccines Internationally. She made it up, but it's nice, right? I like that. Global Alliance for Vaccines and Immunization. There you go. And yes, Destroy U.S. Aid and now Gavi. I think, Gavi gets contributions from many countries. I think they can make it up, but there's no reason for him to stop that funding. He is going to take people's lives as a consequence, and he has no compunction about doing that whatsoever. No conscience. Yes, Kevin says only 7 showed up to the ACIP. 1 backed out because of financial conflicts, and he didn't want to give up his investments. Now Redwood will join FDA CDC to explore and PHAC all HHS patient-level data sets for September bombshell. Great. Hit the like button, folks. Yep. We have 160 watching, 143 likes. Hit the like button, please. It's very easy. Why is Bill Gates not speaking out against unlawful and horrible anti-vaccine stances of RFK Jr.? It's a good question. I don't know why he's not speaking out. Let's look it up. Well, he has warned that RFK is misleading people. He's expressed concern about his decisions about, cutting funds in poor countries. And so he has been kind of, low key, but he has criticized him. Maybe not as hard as he should. So Jill offers to share a New York Times gift link if anyone needs access to an article. Thank you, Jill. That's very kind of you. I'm with Nevitz. Sandy says the microplastics may be making us sicker than viruses. Right. But viruses are making us sick, and we need to vaccinate. Okay, there's no question about that. A couple of more terms for post-infectious immunity. We have wild immunity. That's interesting. Roulette immunity. That's good. Yeah. Yeah. Post-infection. There it is. Post-infection. So Noir had suggested post-infectious. But I mean, post-infection is good. I like that. Post-infection. Maybe that's what Noir suggested anyway. Post-infectious infection immunity. MN, thank you for your contribution to science communication. Thank you, Microbe TV Twiv, for informing my decisions on to get Paxlovid the time I had COVID. Awareness matters. Survivor's immunity. Well, you may not survive, right? Well, I guess it wouldn't matter then, would it? Acquired immunity. Well, that's a general term for the B cells and T cells that make antibodies and CTLs, right? That's a name for that. So we can't use that twice. Yeah. Nevin said, by the way, I thought of a way to describe herd immunity, not herb, but herd immunity while standing in the rain during the No Kings March in New York. Not everyone needs to open an umbrella, get vaccinated to cover 100% of the marchers. Yes, that's very good. I like that. So Patricia thinks Gates is focusing on vaccines in third world countries, hasn't spoken out about the politics of infectious disease control very much in the U.S. Yeah. Isn't the administration trying to make asbestos great again? MAGA. Viruses Luna says, I have a local immunodeficiency in my lungs and I'm happy vaccines helped me not die. If I happen to see, Sharon says, someone wearing a mask, I immediately assume they are immune suppressed. Live and let live. Just take good care of ourselves always. Well, they may be, or they may just not want to be infected. Maybe they have a normal immune system, but they don't react well to respiratory infections, right? Could be a number of things. Patricia has used infection-induced, but post-infectious immunity is good too. Yes, SL says all immunity is natural. One can distinguish post-infection from vaccine-induced. Yeah, well, we want to get away from natural, right? Because then, as Mark Martin said, that makes people think, oh, it's the right thing to do. I don't have to get vaccinated. I want natural immunity, right? Oh, angry was not a real word. That convalescence. Okay. Actually, there's a definition online for it. Why is the SARS inactivated vaccine not as effective? I don't know that we know the answer, but they're inactivated with formalin, and I suspect that it just destroyed the spike protein, so it doesn't induce good antibodies. Mike asks, shouldn't pediatric residents get MMR vaccine regardless of age because of the rubella component? I don't know. It's a good question. If it's required or not. I have no idea. If some physicians here know, let me know. Let us know. Since the USA CDC is now run by anti-vaxxers, what other national agency would you trust for recommendations, e.g. in the EU? Well, you trust the WHO. I think they make good recommendations. But there's some alternative committees being put together here in the U.S. So Michael Osterholm at CIDRAP has already made one, and I hear a few others are getting put together. I mean, how they will do it is a good question because the CDC did a lot of the research leading to the recommendations, right? So I don't know how these alternative ACIPs are going to work. I don't know. More masking during cold and flu season. I don't think I see that here. There's still people masking here, but to the same extent that I see in the winter. Plural of anecdote is not data, but buses in general, the people who sneeze and cough, don't wear masks, but those who do wear them don't cough or sneeze. I think people sneeze in masks occasionally, right? I do. And I just have the mask on and I put my hand over it. In addition, sometimes you can't help but sneezing, right? Yes, the Yamagata strain of influenza B virus has gone bye-bye. We hope, because we took the vaccine component out of the vaccine. Lori says, I had whooping cough when I was 13, freaked my mom when I turned blue and pressed my face against the window screen to get air. 6 sibs didn't get it. Their vaccine worked well. So it didn't work in you. Huh? So tomorrow, the CDC ACIP live stream, day 2626 at 5 a.m. And here's the link for that. Thank you, Les. The expansion of willful ignorance is truly something to behold. So just remember, the ACIP is going to make recommendations, and they're going to have to be followed, I guess, right? Well, it is a recommendation, though, right? So states don't have to follow it, but for insurance purposes, it gets a little tricky there, right? Visto went to Hendra and survived. Well, it's a pretty rare infection, Visto. You're not likely to have got it. Yes, flying foxes are big, as Visto says. Their wings can reach from one electrical wire to the next. Fried bats on the power poles. Are you being serious or are you being funny? Because I know you are funny sometimes. The wires are that far apart, or I thought they were pretty close. I don't know. During COVID, Angry Penguin says, my boss asked me to source an air filter for the office. He was upset when he saw I ordered HEPA filters, thinking the mesh size was too small. HEPA is the right way to go. F. Yang says the most memorable thing of Nipah virus is that the RNA-dependent RNA polymerase looks the most like a certain thing among all the protein structures I've ever seen. You mean it looks like RNA polymerase should? Polka Dot Pup in Austin, hundreds of people gather on a bridge at dusk and wait for tons of bats to fly off from the evening. I was one of them. Can you imagine how much we were exposed to? I went there once and did the same thing. I sat on the bank of the river and watched these bats swirling around for a while, and then they're all gone. It's very quick. And a guy behind me said, is that it? Is that it? But yeah, there's a lot of aerosol going on there, and it just shows you how rare these spillovers are. By the way, if you want to hear more about emerging viruses, the full lecture is on my Columbia University course on YouTube for free. John, thank you for your support of science communication. Lots of Microbe TV episodes this week. So here's my contribution to help keep the lights on. Thank you, John, and everyone else who has contributed. Yeah, we did have quite a few this week, didn't we? That's the link for the emerging viruses or the virology course. John says, date palm sap reminds me strangely of Dr. De Palm. Yeah, we probably talked about it a lot. He was into that kind of thing. And yeah, David Quammen writes about these viruses in Spillover. Wait, Questella says, they found some dead thing and ate it? They can't hunt? I don't think it's so easy to hunt chimps, especially gorillas. You can't find them at all. maybe you don't eat any dead thing you just found. Yeah, but people are hungry. There's no supermarket to go in and get your meat, okay? So we have to cut them some slack. Vista says, don't eat dead chimps. Actually, never eat chimps. They are people, too. 98% human, right? Dead animals don't run and can't physically hurt you. If you are hungry, you eat them. That's it. Yeah. F says there was a news I remembered like 20 years ago. Someone found a dead chicken on the ground in China and took it home. Some toxin in it killed all but one of the family. You should not eat roadkill. No. SIP3, Vincent, your influence on me inspired my girlfriend to buy a microscope, and now we are involved in an ongoing war using predator mites from California to eat bad houseplant bugs in Sweden. Well, that's really interesting, but I'm glad. I'm glad. Oh, you have photos. Cool. I'm glad you got inspired. You want to send the photos to, this is predator mites. Well, you know, you could send them to twiv at microbe.tv. We'd like to see them. And Nevitt says, we must have an organizations like USAID and pandemic teams, etc., fully staffed with experts ready to go immediately to do the epi to head these viruses off at the pass. Totally. Totally. Questella, have you watched Contagion? If not, you should watch it. It's really good. It goes with the emerging viruses lectured. Because that virus in Contagion is kind of loosely based on Nipah virus. Mark got his first COVID after 5 years. It's a timely mini lecture. Good. I hope you're okay. Bobby says, isn't it possible for a MERS human infection or similar to result in a mutation that makes MERS more infectious and then we're off to the races? Yeah, it's possible. That's the worry, of course, right? That it sustains a mutation by just random mutations and then that helps transmit. That's why we want to be careful. And, you know, we should have antivirals or vaccines available for the camels, the vaccines, but antivirals would be great for people too. Yes, I totally agree. F wants to know why so many non-orthomyxoviruses minus ssRNA viruses are so deadly. All the others are nowhere in the 50% plus mortality rate in so many of these viruses. Yeah, I don't know. We got Nipah and Hendra and Ebola, right? But then, yeah, SARS-1 was less than 50%, but that's a plus strand. And what else? Are there any others in the negatives? I'm sure there are, but I'm not thinking of them. Will remembers visiting my grandmother's grave in the 60s and always wondered why there were hundreds of child graves. Thank God for vaccines and pharmaceuticals. Well, let's hope we don't return to those times, right? With taking vaccines from us. Ridiculous. Dr. McFarland, I'm glad you liked it. I appreciate it. It's a lecture that I've been working on for 12, 14 years, yeah. The tacky compactor Barrack said it. He said you can't rule it out, but maybe, I think he said that before the evidence for the market emergence came out, which is very strong, and he probably wouldn't say that anymore. But no, don't give it a 50-50 chance. There's no evidence for a lab emergence. There's evidence for natural emergence. Yes, Les says the banal name derived from the fact that the bat anal swabs are used to collect the samples. Less teeth on that end of the bat. Right. Yes, bats live and fly in huge communities, and bat species mix in caves, for example. So there's a lot of opportunity for cross-infection. Yeah. Patricia, hello. Sorry you're not feeling good. Feel better. Pisto says the bats fight with the possums in the trees, and the possum poo is everywhere in my yard. I'm worried now. It's just, you have to stay away from it, right? Are not pandemics increasing in frequency? Not really. So we have 1968 flu. We have 1977 flu. We have 1981 AIDS. We have 2009 flu. And then, of course, SARS-CoV-2 in 2019. So the thing is, before the 1900s, then we have 1918 and 1957 flu. There probably were pandemics, but we didn't know about them, right? And they're not recorded. So I think, are they increasing in frequency? You don't have enough of a sampling number. You would like to think so, because you have all of these clustering since 1957, right? And the population is increasing. We're doing all this anthropogenic stuff that increases spillovers. I think, though, that we can't say because we don't have enough data from previous years. It's a good question, though. It's a really good question. I like it. Don't eat possums, Visto. I'm going to look in here. We have 3 minutes. I want to see if there's anything else here. Patrick, this is in Puget Sound, 67S. F. Loving it, and extend our condolences to all who are suffering in the horrific heat. He probably got sick of everyone blaming the pandemic on him. He's just talking about Ralph Barrick. That's very funny, Christella. I like that. You're going to be Vincent for Halloween? How are you going to dress up? Do you just get a pair of red glasses? That's it? I don't know. That would be interesting. Yeah, 2 words. 2 words. Post-infection immunity. So post-infection is 1 word. Immunity is the other. So you got it. I like the post-infection immunity. Prions are purely protein. It took 20 years to fully prove and convince the scientific world that it was only protein that was infectious. And it's a weird conformational change. Yes, and in fact, we have a lecture on that in the course. Coming up. Legotic is going to be Paul Offit for Halloween. That's cool. Unplanned immunity. That's funny. The lecture is great. If lectures could be copied to their own videos and on a playlist, they would be more accessible. I could do that, but I have my lecture courses, which are basically the same thing or similar. I don't know if it's worth doing that. The take of today: don't eat dead monkeys you find in the forest. Yeah, a lot of people are liking post-infection immunity. You make it 1 word, post-infection, 1 word, right? Elizabeth really likes the alternative ACIP ideas. Okay, there are a lot of good questions here, but we're going to have to wrap this up. Let's just scroll down here and see if there's anyone I have to thank. Oh, here's Veronika. Hello, Privyet. Check it out tomorrow. Stay safe, Privyet. Legata came up with a new name for my punk band, which does every evening, and today is bat anal swabs. Oh my gosh. It's funny. Okay. Vanity Nutrition gets the final chat of the day. My dogs know more about science than RFK Jr. I start the Virology playlist when I leave for work. Oh, you leave the dogs home and they listen to Virology. That's brilliant. All right, folks. Thank you so much for being here today. Let me thank our moderators and everybody here today. I don't even have to look at the list. We had Vanity Nutrition, Les, Tom, Barb Mac UK, and Steph SF. We have 5 mods. They're all here tonight. Thank you so much for being here and making this civilized. Thanks all of you for coming as well. Great questions, great banter, great chat. When are we going to be back? Let's take a look. At the schedule, office hours, Wednesday evenings, 8 p.m. Next week is going to be, oh my gosh, July 2nd already. That's it for June, folks. And that can only mean the summer is 1/3 over. I consider the summer June, July, and August because those are the best weather months. So, boom, 30% gone, folks. Anyway, Wednesday, July 2nd, we will be back with another office hours. Until then, let's see. Love you all. Stay safe. Thanks for coming. Bye-bye.